Okay, good evening everyone. Good evening Abdul, good evening Annie. Okay, so friends, in the yesterday class, we have discussed about the most important thing, that is the carbohydrate metabolism as well as the process of regulation of the carbohydrate metabolism. Is it? Good evening everyone. So friends, now in this particular class, we are going to study about the another very important topic, friends, that is the amino acids. And I told to all of you friends, from the amino acids you can expect the part B question also, friends. Even you can expect the part C question also. So what I want you people to understand is that in the amino acids, the amino acid topic itself very important for the CSIR exam, either for the 2 marker or for the 4 marker question. But while reading this particular, good evening Shafia. While reading this particular amino acid topics, friends, good evening Anuj, I am Anju. I am requesting all of you read each and everything without leaving any kind of small concept also. Okay, so now let's try to understand. Okay, so from where, good evening. So from where we can expect the questions? So everyone remember my words. We can expect the questions majorly from their pKa values. Friends, we can expect the questions from the isoelectric point, and another very important point, friends, we can also expect the questions by linking pi with pH. Good evening Vinita. So by linking the pi with pH also they can raise the questions. Hence my friends, it is very important you people has to read each and everything without fail. Okay, so do not miss any concept under any circumstances. Okay, now let's start amino acids. So friends, please let me explain for 20 minutes. After that I will solve all the doubts. Okay, so listen to me continuously, friends, for the period of 20 minutes. We all people know very well that whatever the function performed by the body, everything is under the control of a protein. In single line, every student please listen my words, sir. In single line, 37.2 trillion cells present in our body. Once again, every student remember, 2 trillion cells present in our body. Whatever the function performed by that these particular cells, it is under the control of a protein only. Any function. So let's consider, friends, we all people know that the oxygen can be transported in our body continuously to all the cells and tissues. Good evening, by the hemoglobin. Friends, good evening Harsha. What is hemoglobin? Obviously protein. And friends, you all people know very well, if the brain and nerves wants to work continuously, friends, there will be a continuous movement of the ions like sodium, potassium, calcium, all these. And friends, how does this particular ionic movements can be mediated? Good evening. The ionic movement can be continuously mediated by, friends, channels and carriers. Do not get confused, friends, by the channels and carriers. What are these channels and carriers, friends? Obviously they are also a proteins. And please listen my statements. The enzymes, can you imagine our body reactions without enzymes, friends? No, it's not even possible. We can't imagine our body reactions without enzymes. Friends, what are our enzymes? Of course, enzymes are chemically protein in nature. And friends, if you take an example of a hormones are also chemically, most of the hormones chemically proteinaceous in nature. Take an example of insulin, take an example of glucagon. And friends, even if you take an example of cell division machinery, let's take a protein actin, let's take a protein myosin, let's take a protein microtubules. These are all what? They're extremely required for the cell division. But the point is what, my friends? They are all proteinaceous in nature. And let's take an example of a antibodies. What are antibodies, friends? Antibodies are also undoubtedly protein in nature only. Next one, receptors. Friends, I have told to all of you many times. Everyone listen once again. Inside our body, everyone pay attention, friends. Inside our body, if any kind of a function wants to occur, it is compulsory, it is mandatory. What is compulsory and what is mandatory, friends? One or other function, one or other growth molecule, growth factor or anything, if they want to show their action, they have to attach to the receptor compulsory. Without receptor, hormone cannot show their action. Without receptor, growth factor can't show their action. Friends, what are these particular receptors chemically? Undoubtedly, friends, they are protein in nature. If a muscle movement wants to occur, we require a protein. And if the hair, nail, ligaments, tendons, bones, joints, if all these wants to be stronger, what do we require, friends? Undoubtedly, friends, we will be requiring the proteins. Please do not get confused with this particular point. These are all extremely important for the body functions. In a simple word, each and every function performed by the cell, they are under the control of protein only. And friends, how does that particular protein is getting synthesized? That is the valid question. Friends, that particular protein is getting synthesized by what, friends? By the amino acids. Okay, friends, I will teach little bit faster. I want you to catch up it, okay, because we have studied it. So if you look into the structure of an amino acid, we all people know that, friends, it contains central carbon. We all people know that, friends, that central carbon, we always call it as the central alpha carbon. On the right side, it is having carboxyl group, friends. On the left side, friends, it is having the amine group. Then followed by one hydrogen, friends. Then followed by the one R group. Please don't get confused with this particular point. Now, friends, since this particular carboxyl group associated with the alpha carbon, that's why we can call alpha carboxyl group. And friends, this particular amine group is associated with the alpha carbon. That's why, friends, even we can call it as the alpha amino group. So what is the point here? 20 amino acids basically consist of this particular structure. Then, friends, one amino acid can be differ from the another amino acid, friends, exclusively from the R group or exclusively from the side chain only. And friends, everyone please listen my statements very carefully. Do not get confused with this. In the nature, there are roughly, once again, every student remember, in the nature there are roughly 300 amino acids are present. Once again, please listen. In the nature, roughly, friends, 300 amino acids are present. There is no doubt about it. But the point is what, my friends? Out of 300 amino acids, our cells can't use the 300 amino acids for the biosynthesis of protein. Out of the 300 amino acids present in the nature, every student please listen, only 20 amino acids can be used as a building blocks of the protein. Now friends, what about remaining 200 amino acids? Do you think the remaining 200 amino acids present inside the cell, will it be in the inactive form? No friends, whatever the 280 amino acids present inside the cell, they are also required for particular function. The only point you have to remember is only 20 amino acids can be used by the cells for the synthesis of proteins. Remaining, some of the amino acids can act as a metabolites, some can act as a messengers, some can act as a regulator. But the point is very clear friends, every single amino acid is required for the growth and development of an organism. Now friends, please listen my statements once again carefully. Please remember, there are two amino acids, selenocysteine, which is the 21st amino acid, and friends, pyrrolysine, this is the 22nd amino acid. What is the question they will ask in the exam? The selenocysteine and pyrrolysine, which are the 21st and 22nd amino acid, will they really considered as the major, will they consider as the 20 type of amino acids? No. But what is the statement they will ask? What most of the students will make it wrong. They will give the statement, everyone please listen to me carefully. They will give the statement that selenocysteine and pyrrolysine. Once again listen, selenocysteine and pyrrolysine. These two are the amino acids. Everyone remember, they can genetically incorporated into the proteins. This is the statement they will ask. You all people will absolutely give the wrong answer, because the selenocysteine and pyrrolysine are the 21st and 22nd amino acid, so they are not present in the protein, so which is absolutely wrong statement. Every student please listen my statements. Selenocysteine and pyrrolysine, they are genetically incorporated into the proteins. Now you can ask me, sir, does the selenocysteine and pyrrolysine will be present in every single protein? That is wrong statement. They are not present. Good evening Priya, continue. They are not present in all proteins. Not all proteins contain this 21st and 22nd. But friends, what is the statement I can give to all of you? Some proteins will contain this particular 21st and 22nd. Please do not get confused with this particular point. Friends, just now we discussed that every protein consists of a amine group, carboxyl group and hydrogen. What we discussed friends, the protein can be differed from one to other only with respect to which group my friends? Only with respect to the R group. Yes or no friends? Of course yes. Now listen, side chains are R groups. They are vary in structure, vary in size, vary in electric charge. In very simple point, one amino acid can be differ from the another amino acid exclusively based on the R group. Please don't get confused with this particular point. And friends, what is the average molecular weight of an amino acid? Everyone keep it in your mind. Friends, we are using the word average. Okay, we are using the word average weight is friends 110 daltons or 110 daltons. Friends, if you look into our 2022 February question paper, please listen my statement. We got the question directly from the PI. Okay, a kind of a simple question, I can use the word. But what is the another statement that I am giving to all of you is that there are certain chances are there, instead of giving the question from the PI, what they will do friends, they will link the amino acid question even to the protein. They will link the amino acid question even to the nucleic acid also. Okay, so that's why you have to know different type of numerical questions when you are answering this particular amino acids. Okay, so friends, let's focus. A segment of bDNA encoding an enzyme of a molecular mass of 50 kilodalton the estimated length of this segment in the micrometer would be. What you have to understand by that? Everyone listen, don't get confused friends. I will explain this very simple way. Focus. Everyone listen. This is a BDNA. Okay. What friends? This is the BDNA. Now what is the question they asked? So imagine this is a particular segment. Everyone listen friends. This is a particular segment of the BDNA. This particular segment of the BDNA, it is coding. Everyone remember, it contains a molecular mass of 50 kilodalton. What is the question they asked? Friends, what is the length of this particular segment? Very simple question friends. They have already given the molecular mass of a particular segment and they just asked us calculate the estimated length of this particular segment in the micrometer. That is the simple question they have asked. Now they have given the 4 options. We all people know that. In the previous slide I have told to all of you. Friends, average molecular mass of one amino acid is 100 Dalton. This is what we studied. And what is the molecular mass of protein they have given? They have given a protein molecular mass is 50 kilodalton. So which means friends what? 50,000 Dalton. So 50,000 Dalton by 110. Then friends, how many number of amino acids that we are getting friends? We are getting 455 amino acids. Now every student remember, you people imagine entire BDNA, everyone listen my statements very carefully. If you assume all the nucleotides present in the BDNA are expressed into enzyme, all the nucleotides present in that particular BDNA are expressed into enzyme. What do you have to write friends? So 455 amino acid into 3. So why 3? Because we all people know that. Why 3? Because three bases come close together, they can form one codon. That one codon only code for the one amino acid. We all people know that. Then friends, how many number of nucleotides we will get friends? We will get 1364 nucleotides. We all people know that very well. And friends, you all people also studied that one turn. See here everyone remember, this is called one turn. Correct na? One turn of the BDNA consists of 10.4 nucleotide. Only one turn. How many nucleotides we are having friends? 1364 nucleotides are having in entire DNA. So 1364 divided by 10.4. Then how much we will get friends? We will get 131.2 turns we will get. We all people read in the chemistry part. Length of one turn. Length of one turn is how much friends? 3.4 nanometer. This is a fixed, right? This is a fixed. So friends, then 131.2 turns into 3.4. How much we are going to get friends? We are going to get 446 nanometers, or 0.4641 into 10 power minus 3. This will be the answer. I will re-explain it in a very quick way. All the students listen my statements carefully. This is the DNA. They have given the weight or a molecular mass of a small segment which consists of the 50 kilodalton. And what they asked friends, what is the estimated length? They just asked us the length. So since they have given the mass, first we are identifying how many number of amino acids are present. So they have given the mass of 50,000 Dalton, and we all people know that the average molecular weight, average molecular mass of an amino acid. Then I can easily calculate the total number of amino acid. When I calculate the total number of amino acid, every student please remember, when I calculate the total number of amino acid, I can easily calculate the total number of nucleotides that are present. 455 into 3. Why we are taking 3? Because the triplet is the one which is going to code for the amino acid. We got 1364 nucleotide. We all people know that, friends, one turn of a bDNA, because it will be varied from DNA to DNA. ZDNA different nucleotides are there in one turn. aDNA contain different type of nucleotides. So friends, 1364 by 10.4, we will get 131.2 tons. So finally we got it. How many number of tons? But the question asked is, they did not ask the question how many number of tons. They have asked the question what is the length. So we all people know that length of one turn is 3.4 nanometer. So we will be multiplying it 131.2. So whatever we get is our answer. Most of the times they won't ask this kind of questions, most of the time. But sometimes they will link these questions with the amino acid, then they will ask. If you only know the molecular mass of amino acid, and if you really don't know about the length of that particular one turn in the bDNA, friends, you can't answer this particular question. So that's why, friends, it is very important you should know each and everything. Okay, now, so that is a simple numerical. I hope every student understand it very correctly. Now, friends, tell me. So suppose example, everyone listen. This is the amino acid, having a carboxyl group, having a amine group, having one R group, and having one hydrogen. This is the simplified structure. Friends, what I have done, I have dissolved this particular amino acid in the water. When I dissolve the amino acid in the water, what is going to happen? Friends, this particular COOH lose this particular H. Correct na? Upon losing, it is converting into what, friends? COO minus. Now, who is going to accept this hydrogen? This hydrogen can be accepted by NH2, friends. Then it is converting into what, friends? NH3 plus. Correct na? All the students, please keep it in your mind. COOH and NH2. These two are called as what, friends? These two are called as the non-ionic forms. Whereas COO minus and NH3 plus, these two are called it as what, friends? Ionic forms. So always remember this particular point without fail. Okay. Now, why I am talking about this particular non-ionic form and ionic form? Every student has to remember, when we are calculating the pKa value, we should always consider the values of the ionic forms. Friends, I don't know what you do. I think you have already remembered this. If you did not remember, I am requesting all of you, you have to remember compulsory. What you have to remember the compulsory? What is the form of this particular ionic forms, what is the charge of ionic forms at the physiological pH? Once again, I am reminding to all of you, friends, what is the charge of ionic forms at physiological pH? It is compulsory, it is mandatory, you people has to remember, there is no doubt about it. The alpha carboxyl group, please remember here, nothing to explain, having the low pKa value, right? And please remember, friends, so what is the point you have to remember? What is the physiological pH, friends? At the physiological pH is 7. So now you can understand this COO minus this COOH at a physiological pH 7, what happened? It will get deprotonated. We all people know that, friends, if it is a deprotonated, what kind of charge it acquire, friends? It will acquire the negative charge. You have to remember it compulsory. Whereas, friends, what about this particular alpha amino group? The alpha amino group having a high pKa value 9 to 10. Hence, friends, therefore, it is protonated. And friends, it is positively charged. Friends, it is protonated as well as the positively charged at a physiological pH. Correct na? So, if it is a protonated, positively charged. If it is a deprotonated, negatively charged. I don't know, friends, what you have to do, but I want you to remember it compulsory. Okay. Friends, now let's take human beings. Let's take, friends, human beings. Let's take a human beings. If you isolate or if you take the protein out of a human, when you observe, friends, you can get most of the amino acids or L amino acids only. This is a very important, friends, which means that most of the amino acids are rotating the plane polarized light towards the left hand direction only. That's why, friends, we will be calling it as the L amino acids. Please don't get confused with this particular point. Now, what about the bacteria, which is they are already asked in the exam? Most of the bacteria, the amino acids are, friends, D amino acid, dextrorotatory, which can rotate the plane polarized light in the right hand direction. Example, everyone please remember, example, most of the proteins that are present in the bacterial cell wall. Please remember that without confusion. Okay. Now here comes another important. Is there any necessity to remember the classification of amino acid? Friends, not only for the CSIR exam, even for the ICMR, DBT, GATE, XL, for any kind of an exam, it is compulsory, it is mandatory. Amino acid classification you have to remember, and it is very important and it is very mandatory. You have to remember 20 amino acids structure. You have to remember 3-letter codon. You have to remember single letter codon. It is your least responsibility, at least. Okay, so do not miss at all. When compared to the 3-letter codon, my friends, it is always important to remember the 1-letter codon. Okay. Now, sir, listen my statement. Apart from the numerical, please listen. Apart from the numerical, what are the other chances are there that we can expect the question? Friends, other chances are there. Please remember, we can get the questions from properties. Good evening, Saima. Please continue. We can expect the questions from the properties. So my friends, remembering the properties are also extremely important. I am requesting all of you, friends, not to skip this particular properties. Okay, what kind of properties, what class they belongs to, and most important, what are the functions performed by them? Remembering their functions is utmost important, friends. Okay, now let's focus. Non-polar aliphatic R groups. What you have to understand by the word non-polar aliphatic, friends, you can see the picture, right? The amino acids almost, they are all in the linear forms. Alanine, valine, leucine, isoleucine, glycine, methionine, proline. You have to remember. Aromatic R groups, which are branched. Phenylalanine, tyrosine, and tryptophan. We all people know that the hormone auxin in the plant body, it is derived from the amino acid precursor, which is the tryptophan. The most important one, uncharged R groups. Serine, threonine, cysteine, asparagine, followed by the glutamine. Right. Now, positively charged, which are basic lysine, arginine, histidine. Friends, every single amino acid having their unique importance. It is your responsibility to remember their unique importance. Negatively charged are aspartate as well as, my friends, glutamate. Don't get confused with this particular point. Okay. Now listen. Non-polar aliphatic R groups. I will explain one by one. It's easy to remember structure. We all people know it. We studied many number of times from our 11th standard. Glycine is the most simplest amino acid. Why glycine is the most simplest amino acid, friends? Because the R group just contain hydrogen. Except hydrogen, the R group do not contain anything. And we also know very well, glycine is the only amino acid which is optically inactive because it do not have a chiral carbon. So it cannot rotate the plane polarized light in any direction. It can't rotate in the right hand direction. It can't rotate in the left hand direction also. Friends, now you only try to understand. Please remember in this particular short method, alanine. Alanine contain what group? You can see, right? Alanine contain methyl group. Very simple. And friends, what about the valine? All the students look into the diagram. Friends, this everyone look into this. This valine contains same CH3CH3, just there is a addition of CH. Now don't change any structure. Keep this entire structure and just add CH2. Very simple, right? And one more very unique one. The moment you heard the name of a methionine, you can easily remember that there is a presence of the sulfur. And when you heard the isoleucine, just remember that to this particular structure of a leucine, you have added another extra carbon. That's all. No difference at all. Once again, I am telling you how to remember the structure. Because friends, you have to remember it. Glycine contains simple hydrogen. Alanine contain methyl group. Valine contain methyl groups are added with one CH group. The same structure of valine added by the another CH2. That is called leucine. And the same structure, it is just added by the extra carbon. That's all. That is the isoleucine. Whereas methionine is what, friends? Sulfur containing amino acid. I want you people to remember it compulsory without fail. Glycine has a single hydrogen atom. Alanine has a methyl group. They are all having branched aliphatic side chain. Sulfur containing. And friends, we all people know that proline. The moment you heard the name of a proline, two statements every student has to remember. Three statements. What is the first statement? Proline is the helix breaker. You all people know that. Second one, proline is rigid. This is also fine. But friends, what is the third point I want every student has to remember is that proline is a very good amino acid in the starting of the protein. It's a very good amino acid in the starting of a protein. In the starting of a protein. Friends, please do not get confused with this particular point. This is a really unique statement. What is that? Proline is a very good amino acid in the starting of a protein. So please don't think that if you keep the proline, suppose example, this is the helix. If you keep the proline here, it will break the helix. But friends, if you keep the proline here, friends, it is really helpful in providing the rigidity to the helix. By that, the helix will become extremely stronger. Because you can ask me why. Because in the starting, proline will provide the strong covalent bond. Since it provides every amino acid will provide the covalent bond. But in the starting, proline can provide the strong covalent bond. That's why, friends, we can't break it very, very easily. Now, aromatic amino acids. Easy to remember, right? It contains phenyl group. That's what we can call phenylalanine. And it contains what group? Phenol group. That is what we can call, friends, tyrosine. And friends, we have studied about the tryptophan. What kind of a ring tryptophan contains? Indole ring. Friends, remembering the structures are not at all difficult. To be frank, it's very, very easy. Okay. Now, what are the unique statement every student has to remember for the exam? Friends, this particular tyrosine, which is having OH group. Everyone remember this. This oxygen, in most of the cases, it is acting as a nucleophile, which is involved in the nucleophilic attack. Please remember this particular point without fail. And friends, tryptophan. Tryptophan is the amino acid which is acting as a precursor molecule for many kind of a amino acids. Now, what about absorption of light? Please listen my statements very carefully, friends. When compared to the phenylalanine and when compared to the tyrosine, friends, tryptophan is the amino acid which can absorb the light at higher intensities. So if you look into the phenylalanine very specifically, it can absorb the maximum amount of light, 260 to 270 nanometer. If you look into the tyrosine, it can absorb 280 to 290. But friends, if you look into the tryptophan, it can absorb in between 300 to 310 nanometer. They can ask a question, which of the following amino acid can absorb the higher intensity of light? Okay, obviously remember friends, tryptophan. Do not get confused with that point. Now next, polar uncharged R groups: serine, threonine, cysteine, asparagine, glutamate. Friends, easy to remember, right? The moment you heard the name of a asparagine and glutamate, what you can remember? Asparagine contain extra amine group, glutamine also contain extra amine group. Easy to remember. And friends, I will explain about the cysteine separately. Please remember, serine and threonine, these two are the amino acids which plays a key role in the process of the phosphorylation. It's a very important. Let's take an example of a RNA polymerase enzyme which is involved in the transcription of a eukaryotes. So if you look into the structure of a RNA polymerase, in the C-terminal domain of the RNA polymerase, friends, there is a presence of amino acid serine, and friends, there is a presence of amino acid threonine also. We all people know it very well. Now please listen, the amino acid serine and the amino acid threonine both are getting phosphorylated by a specific kinases. Correct na? So what? And not only that, there are certain proteins, by using the serine, please remember friends, they will undergo a process called as O-linked glycosylation. Friends, we all people studied many number of times. Friends, if the protein wants to be biologically active, everyone listen, if the protein wants to be biologically active, the protein has to be compulsory added by the sugars. So how that sugars can be added? The sugars never added randomly to the protein. That is also wrong statement. Sugars can be added to the proteins at a particular area, which is we can call as O-linked glycosylation. There are some proteins are there, they will also added by the sugar with the help of a N-linked glycosylation. And we all people know that the O-linked glycosylation is taking place in the Golgi complex, which you have to remember compulsory. And friends, this N-linked glycosylation is exclusively occurring in the RER lumen, which you people has to remember compulsory. Do not get confused with the points. The more information you remember, more easily you can answer the questions. Okay, but what about the special amino acid cysteine? Please, every student, please look into the diagram very carefully. You can see, right, cysteine, two cysteine molecules. This is the first cysteine, this is the second cysteine. The two cysteines are associated with each other. Correct na? By what linkage? By a sulfhydryl linkage. Okay, everyone remember friends, the two cysteine molecules are associated with each other by the sulfhydryl linkage. Correct na? Now everyone just listen. If it is associated with this particular sulfhydryl linkage, just listen to me carefully. What happens? There will be a process of oxidation. Friends, this also you can expect the question in the part B. Okay, which of the following chemical reaction is taking place in the formation of a disulfide bond? What's the question we get? Which of the following chemical reaction is taking place in the formation of a disulfide linkage? Everyone keep it in your mind, friends. The oxidation. So what is happening, friends? This particular cysteine, the two cysteine molecules undergo the oxidation. Then what they are going to form, friends? They are going to form disulfide bond. So I have already told you, right? If you want to form a disulfide, if we want to form a disulfide bond, how many amino acids required? Two cysteine amino acids required. So based on this, they can raise the question. Suppose example, there is a insulin. We all people know that insulin contain chain A and insulin contain chain B. like this they can ask the question. The chain A contain 12 cysteines and the chain B contain 4 cysteines. Now what is the question they have asked? They can ask a question, total how many number of disulfide bonds that are present in this particular protein, insulin. Here 12 cysteine means how many bonds will be formed? 6 bond will be formed. Here 4 means 2 will be formed. So in this particular insulin, total how many disulfide bonds will be formed, friends? There will be a formation of the 8 disulfide bonds. Like this also, friends, they can raise the very simple questions. Now next, please look into the picture, friends. Very easy it is. Positively charged R groups, lysine, arginine, as well as the histidine. You can see, right, friends, lysine contain an extra amine group. Arginine, please remember, friends, arginine and histidine also most important. Histidine contain a ring called as imidazole ring. Arginine contain a ring called, friends, guanidino ring. I want you to remember compulsory what kind of rings that they are containing. And if you look into the simple structures of aspartate as well as the glutamate, they contain what, friends? Extra carboxyl group. I don't know what you do. Okay, so before going to the exam, my sincere request to all of you, friends, sincere request, just read this particular slide. In this particular slide, friends, the 20 amino acids are present. Everyone listen. In this particular structure, 20 amino acids are present. Then, friends, followed by the 3-letter codons are present, 1-letter codons are present, and structures also present. I want you to remember. See how easily I can remember. How easily I'll remember. Glycine, hydrogen, alanine, methyl, alanine, valine, leucine, isoleucine. I can easily remember these 4 structures because they are the aliphatic. Methionine I can remember sulfur, tryptophan I can remember indole, phenylalanine I can remember phenyl, proline I can remember imino group, cysteine I can remember sulfhydryl. Easily by looking at the structure I can tell you. Arginine I can remember guanidino ring, histidine I can remember imidazole, lysine I can remember extra amine, aspartic and glutamic I can remember extra carboxyl group, and serine and threonine I can serine, threonine, tyrosine I can remember OH group, asparagine and glutamine also I can remember extra amine group. I can remember the structures very easily without having any kind of a confusion. That's why while remembering the structures, always remember with the simple shortcuts. Okay, now let's come to the numerical part. So that is the calculation of the PI, which we are expecting the questions. But I am requesting all of you, while calculating the PI, friends, my sincere request to all of you, focus on the calculation with respect to the pH also, because you can easily calculate the PI just based on a simple formula. But I want you to remember compulsory the PI calculation with respect to the pH. That is important. Okay. Now, what do you mean by the word Zwitter ion? Now, you can ask me a question. Sir, the question is, friends, does the amino acid can exhibit as a Zwitter ionic form every single time? No, I never say that. An amino acid can be exhibited as a Zwitter ionic form only when it is dissolved in the water. If an amino acid is not dissolved in the water, you only tell me what do you call it, that amino acid? Friends, that amino acid which is not dissolved in the water, you will be calling it as the non-ionic form. Very simple point. Non-ionic form amino acid never ever act as a Zwitter ionic form. Zwitter ion, when amino acid dissolved in the water, then because it can act as either as a base or as an acid. Since the amino acid can acting as a base or acid, we will be calling that as the amphoteric compound. What do you mean by the word amphoteric compound, friends? Amphoteric compounds are those type of compounds, friends, which are having the dual nature. That's why it can act as a both acid as well as the base. Please do not get confused with this particular statements, friends. Now you only look into this. Is there any dissolving in the water here? There will be a no dissolve. What do we call it as the uncharged structure? Here I will use the word dipolar because it is having the basic also, it is having the acidic properties also. That's why we can call it as a dipolar ion, friends, or are the Zwitter ion. So friends, once again you people has to compulsory remember, when you can called an amino acid or a Zwitter ion is an acid. Every student please remember, we can call it as an acid when it is acting as a proton donor. We are calling it as a base, friends, when it is accepting the proton. Correct na? Proton donor is the acid and proton acceptor is the base. And these are all possible when the amino acid is dissolved in the water. Always remember that particular point. Okay, now we all people know easily to calculate the isoelectric point. You know friends, in order to calculate and in order to calculate the isoelectric point, see, if you look into your 2022 February question paper, they have given the pKa values in the exam. They have given the pKa values in the exam and they asked you to calculate the pI. But please don't think that every single time they will give the pKa values in the question and they will directly ask you to calculate the pI. Sometimes they don't give the pKa values. So it's your responsibility to remember that particular pKa value. The pKa value of a carboxyl group, we will be calling it as the pK1. And the pKa value of an amine group, we will be calling it as the pK2, because we also discussed that some amino acid, even R groups, please follow me correctly, some amino acid R groups, they are also ionizable. What do you call them as? They are also ionizable. If they are ionizable, my friends, they will be also having compulsory particular pKa value. What do we call it? That as we will be calling it as the pKr. So the pKa value of the R group, we will be calling it as the pKr. Always remember pK1, pK2 as well as the pKr. Don't get confused with this. And I don't know what you do, friends. It is your responsibility. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I am requesting all of you to remember this 10 amino acids, pK1, pK2 and pKr. Especially focus on the pKr. pKr, you people have to remember only for these 6 amino acid. Friends, you can get a part B question like this. What part B question you will get in the exam, you know? All of the following, everyone listen. All of the following amino acid R groups are non-ionizable except. What's the question you will get? All of the following amino acid R groups are non-ionizable except. So please keep it in your mind what kind of amino acid R groups are ionizable without fail. Please remember cysteine, lysine, histidine, arginine, aspartate, glutamate. So these are all the 6 amino acid where the R groups are truly ionizable. My sincere request to all of you to remember that compulsory. Now, if you don't remember that, friends, you can't answer the questions. Okay, see, because if you remember, you no need to look at the options also. If you remember, you can calculate it. For the confirmation, you can look into the option, you can solve it very faster also. Now you can ask me a valid question. Sir, does all the amino acids are having the same isoelectric point? No, we cannot say that. Every amino acid should not have similar isoelectric point. It is completely different. So everyone listen. How can I define the word isoelectric point? Isoelectric point is the pH. Once again, every student remember. Isoelectric point is the pH of where the positive charge is equal to negative charge. Where the positive charge is equal to negative charge. In other words, how can I define? I can define every student remember, I can define isoelectric point is the pH where the amino acid is the neutral. Very, very, very simple point it is. Now, what do you mean by the word neutral? Which means, friends, the amino acid is not at all migrating in the electric field. Friends, this point is very important. Whenever you are calling an amino acid is a neutral, every student keep it in your mind. You have to remember that it does not migrate. Please remember that. The amino acid is the least soluble in water, does not migrate in the electric field, which is important in the electrophoretic separation of the peptides. Very important, friends. So what will be the simple formula to calculate it? We all people know it very well. The simple formula is Pi equal to pK1 plus pK2 by 2. This is the simple formula. Now friends, you only look into this. What will be the overall charge here? Charge is 0 and plus 1. So the net charge is plus 1, minus 1 and plus 1. So when the plus 1 and minus 1s are cancelled, the net charge is what, friends? 0, obviously. Now here, what will be the charge here? Here the charge is 0. And here what will be the charge? Here the charge is minus 1. So what we have to consider for the isoelectric point? For the isoelectric point, we should always consider the where it is in the neutral form. So where the amino acid is in the neutral form? Here. So obviously we will consider this and this to calculate this particular neutral point. Let me explain you how. And friends, this is also very important. Please remember, very, very important it is. At a pH, this is very important. These three statements are very important statements. Please remember. At a pH above its isoelectric point, always remember what is the charge that is carried by the amino acid. The pH above its electric point, obviously friends, the amino acid having the net negative charge. So if the amino acid having the net negative charge, obviously it is moving towards what, friends? Anode, which is the positive electrode. At the pH below its Pi, obviously the amino acid having the positive charge, friends, and obviously where it will move to? It will move to the negative electrode, which we generally call it as what, friends? Cathode. And the most important, at pH which is equal to Pi, which means the net charge on the amino acid is 0. Correct na? So there will be no movement in the electric field. This is only very important property that we will consider when we are separating the protein from a particular solution. Please don't get confused here. Okay. See here in this particular, see I am teaching a little bit faster because we already done this. But it is, see, doing one time is not important. Doing multiple times only important. That is only the important parameter you have to remember for the CSIR. How many number of times you are revising without forgetting the concept is only important. Look into the picture. Here the blue color graph is indicating the zwitterionic form. I mean to say the blue color graph is talking about the amine group. And you can see this pink color one is representing about the carboxyl group. Okay, now friends, let me give you the conditions first. What will be the conditions I am giving? Please remember, I am giving a condition acidic pH. I have given you a condition of acidic pH and I have asked you the carboxyl group is exhibited in which form. What you can say that? The carboxyl group at the acidic pH, obviously it is protonated. Correct na friends? If it is protonated, what about it? Obviously it is in the cationic form. We all know that very well. Now same carboxyl group, what about the neutral? Neutral, it never be protonated, right? Obviously it is going to lose the proton. So it is in the deprotonated form. Correct na friends? Please don't get confused. I am talking only carboxyl group. I am not talking about the amine group. Please don't get confused with this. Now let me explain you about the amine group. At alkaline pH, what about the amine group? The amine group is obviously neutral. Correct now friends? And the amino acid is going to exhibit in which form? Undoubtedly friends, the amino acid is going to exhibit it in the anionic form. Don't get confused. Now I have very clearly told to all of you that friend, what I told to you friends, not only the carboxyl group, not only the amine group, friends, R groups also ionizable. R groups are also ionizable. That is the reason. What is the statement I can conclude? I can conclude a statement that different amino acids having different pKa. Please do not get confused with this particular point. It's a very important statement friends. Now calculation is very easy now. This is the amino acid glycine. Okay, so friends, what you people has to remember, what did I tell you? You have to remember the pK1 value, you have to remember the pK2 value, you have to remember the pKr value. If you remember these three, it is very easy to answer this particular PI question. Now you only tell me what will be the charge here. Here +1 and here 0. So the total net charge is +1. Okay. Now here what will be the charge? Here it is a +1. Here it is a -1. So obviously it is exhibited in the 0. Now here it is, what is the charge? Here it is the 0 and here it is the -1. Now what are the values I have to consider? Friends, here it is easy, but when we are discussing the pH with PI, there only you will get confused. Here it's easy. Which ion is which pKa values are responsible for the neutral form? Which pKa values are responsible for the neutral form? This pKa value is responsible for the neutral form. This pKa value also is responsible for the neutral form. So the pK1 value, the carboxyl group of glycine is 2.34 and pK2 value is 9.60. So by 2, then what we are getting? We are getting 5.97, which is the isoelectric point of the glycine. Very easy. Now look into the amino acid glutamate. What we discussed friends, glutamate having carboxyl group, and what I told to all of you friends, the glutamate is the amino acid which is having R group, which is a ionizable R group. See friends, I can tell you very easily. Listen my words. When the amino acid R groups is in the ionizable form, we can get 4 forms. Always remember this. Okay. Now you only look into the picture carefully. Now since it is having, see, most of the students will say that since pKr value, the amino acid R group is ionizable, so we have to consider pKr also compulsory. It's not mandatory, is it? It is not mandatory that every time we have to take the pKr value since the R group ionizable. Whether the pKr is involved in the neutral form of amino acid or not. If you understand that, then only we have to consider this. Now you look into this. What will be the charge? +1. Here what will be the charge? 0 and 0. So the net charge is +1. And what will be the charge here? -1. 0 and +1. So +1 -1 cancel, the net charge is 0. And what will be the charge here? -1 +1. -1 +1 cancel and -1. So the net charge is -1. So here -1 -1 0 -1 -1, which is the -2. Now friends, you only look into the picture. Which will be the neutral form? This will be the neutral form. Now you have to think it correctly. In the formation of a neutral form, yes, Harsha, that's a very good point. Every ionizable form of an amino acid having a 4 forms. That's a good point you have to remember. Correct. Now listen. Here, this is the 0, which is the neutral form. Hence what you have to remember? -1 plus 1. So you have to remember, should I really consider the pKr value? Should I consider the pK2 value? Now we never consider the pK2 value. What will be the reason why we did not consider the pK2 value? Because the pK2 value is not involved. Every student remember my words. The pK2 value is not involved in the formation of this Zwitter ion. Since it is not involved in the formation of a Zwitter ion, we no need to consider. What we will consider? We will consider the pKr and pK1. These two are compulsory involved in the formation of this. So pK1 plus pKr by 2. Instead, I can also tell you one method. There are majorly 6 amino acids. Major, I am talking major. 6 amino acids where the groups are ionizable. You can also remember the formula. In these 6 amino acid, in order to form the neutral form or a dominant form, whether pK1, pK2, pKr, which is involved, if you remember or by heart these 3 also, without looking at the answer, you can easily solve the question. This is also simple way. Why I said 6 amino acids majorly? Because in a given protein in the question, most of the time they will rise this 6 amino acids only. So you remember this 6 amino acid, what forms are involved in their ionizable group. Based on this also you can easily answer the question without looking the options. Okay friends, is it clear everyone till this particular slide? Okay, so in the tomorrow class we will continue this. In the tomorrow class itself, I'll finish the proteins also. Okay. After proteins, we'll immediately go to the enzymes. Okay. Protein, we will majorly see the alpha helix as well as the beta pleated structures. Primary, secondary, tertiary, quaternary. Okay. Revise it without fail. And friends, that is September 2nd. From tomorrow onwards, the mock test will be starting. My sincere request to all of you, even though you don't given the chapter wise test, no problem at all. I want you to give compulsory mock test. There are 145 questions are there. Part A, Part B, Part C. It is exactly like this particular CSIR paper only. 20 questions, 50 questions and 75 questions. I am sincerely requesting all of you. If you give the 6 test, so 6 into 75 part questions you can read, 6 into 50 part B questions you can read. Okay, so here only you can cover more than 600 questions. I am requesting all of you, please give the mock test without fail. Okay. Okay friends, once the test become live, immediately I'll inform in the telegram. Okay friends, I'll meet you in the tomorrow sharp 4 p.m. We will continue this and we'll finish the proteins. Okay, bye bye, thank you all.